You've found Dreamtime Science, a peaceful corner where we unravel the mysteries of our universe one whisper at a time. Let the gentle current of discovery carry you towards sleep. You know what's fascinating? I've been thinking a lot about how something so tiny, bacteria, actually holds the keys to so much of life on Earth, even though we can't see them with our eyes. There's something remarkable about this invisible world quietly working behind the scenes, shaping everything from the air we breathe to the food we eat. I can't wait to share some of the surprising ways these tiny creatures are not just living but truly running the show in their own special way. If these gentle explorations bring you comfort, a quiet like or subscription would mean the world to me. Your support helps keep these peaceful journeys flowing. Take this moment to settle in, wrap yourself in comfort, release the day's weight from your shoulders, and let your breath become as gentle as starlight. Close your eyes and allow your thoughts to drift, lighter than air, through the porous fabric of the world around you. The room in which you lie seems so still, so solid, a sanctuary of walls and warmth, the gentle hush of distant traffic, the rhythm of your own breath. Yet, hidden beyond the boundary of sight, there pulses an invisible kingdom, ancient and tireless. This kingdom is everywhere and nowhere, on your skin, in your lungs, woven into the dust motes that dance in the slant of a bedside lamp. It is the domain of the bacteria. To most, these creatures are nameless, faceless, shadows that haunt the edges of our awareness. The word bacteria itself shivers through the popular imagination like a ghost story told in the flicker of candlelight. For centuries, and especially in the bright, anxious glare of the modern age, bacteria have been cast as the villains of human life. The unseen assassins lurking in the cough of a stranger, the taint in a glass of water, the tiny saboteurs that might upend the story of a civilization with a single, feverish pandemic. Consider the stories we tell about them. In the world of science fiction, bacteria are often the harbingers of doom, the ultimate plot twist that needs no face to inspire dread. In the classic film The Andromeda Strain, a mysterious microorganism from space threatens to erase life on Earth, its very simplicity making it all the more terrifying, unstoppable, unknowable, immune to the weapons and wisdom of humankind. Or think of outbreak narratives, from Hollywood blockbusters to pandemic thrillers, where the true antagonist is not a villain with a name, but a force that multiplies invisibly, indifferent to borders and allegiances. The image of the germ, the bacterium, is the perfect foe, invisible to the naked eye, yet capable of toppling empires. Even outside the cinema, this narrative has seeped into our daily rituals and anxieties. There is a certain choreography to modern cleanliness, a dance of avoidance and eradication. Hand sanitizer in every purse, antibacterial soaps lined up like sentinels by the kitchen sink, wipes with promises of 99.9% effectiveness. Each product, each label, whispers the same story, that we are beset by invaders, and our only hope is vigilance. The language of warfare pervades these messages, fights germs, kills on contact, eliminates bacteria. In these battles, bacteria are never allies, never neighbors. They are always the enemy. Yet this view is a recent invention, born of a century's worth of scientific discovery and cultural transformation. To the ancient world, the invisible was not microbial, but mystical. Illnesses were the work of spirits, humors, or curses. The notion that tiny, unseen creatures could shape the fate of kings or the health of a child was unthinkable. It was only in the 17th century, when Antonia van Leeuwenhoek peered through his handmade lenses, that humanity first glimpsed the teeming world beyond our senses. He saw animalcules, tiny motes darting in a drop of water, and marveled at their abundance and variety. But it would be generations before these creatures were recognized as agents of disease. or indeed as anything other than curiosities. The germ theory of disease, which finally took hold in the 19th century, was as revolutionary as Copernicus's heliocentric model or Darwin's theory of evolution. Suddenly, the world was reimagined, not just as a landscape of visible threats, but as an intricate battleground where every surface, every breath, every mouthful of food might harbour invisible armies. The microscope became a kind of magic mirror, revealing monsters where none had been seen before. And so began the age of antiseptics, of sterilization, of the germ-free ideal. This newfound awareness reshaped not only science, but the culture of daily life. Hospitals adopted strict regimes of cleanliness. Surgeons scrubbed their hands and instruments, and the rate of infection plummeted. The language of hygiene entered the household, with mothers and fathers instructed to keep homes spotless, children clean, and food untainted. Advertising campaigns for soap, bleach, and disinfectant flourished, their illustrations brimming with cartoonish depictions of sinister, bug-eyed germs. The message was clear. To be modern, to be civilized, was to wage constant war against the invisible. And in this war, bacteria became the catch-all bogeymen, blamed for every malady and every imperfection. The flu, the common cold, the stomach ache, never mind that viruses, fungi, and other agents played their roles. The microscope's revelations, simplified and amplified, turned the microbial world into a singular threat. In the imagination of the public, it was easier to picture a single type of enemy, an army of tiny, identical monsters, than to grasp the mind-boggling diversity and complexity of the microbial universe. Popular media has only sharpened this paranoia. In countless news stories, bacteria are described in the language of invasion and resistance, superbugs that defy our drugs, outbreaks that threaten to spill across borders. The rise of antibiotic resistance is cast as a new arms race, a desperate scramble to stay one step ahead of evolution itself. In the sterile corridors of hospitals, the spectre of MRSA and C. difficile haunts every surface. Even the word bacterial carries a shiver of foreboding, a taint of something not only dirty, but dangerous. Yet, for all this fear, bacteria remain largely unseen. They do not appear on the nightly news in the way a wildfire or a hurricane might. Their work is silent, their movement hidden. A single cough can release millions into the air, but they vanish before our eyes, their passage detectable only by the symptoms that follow. In this way, bacteria are both everywhere and nowhere, blamed for every ill yet rarely recognized for their true ubiquity. This invisibility, this paradoxical presence, makes them perfect fodder for the stories we tell to frighten ourselves. In the fevered pages of pulp fiction or the flickering frames of a dystopian film, the bacterial threat can be anything: a weapon engineered in secret labs, a plague loosed by accident or malice, a punishment for hubris. There is something primal in the fear of contagion, of a force that slips past all defenses, that cannot be bargained with or reasoned into submission. Bacteria, in their multitudes, become a cipher for our anxieties, a metaphor for all that is unpredictable, uncontrollable, unseen. But the story does not end with fear alone. The modern obsession with cleanliness, with banishing bacteria from every corner of our lives, has taken on the fervour of a secular religion. Supermarkets overflow with products promising ever greater purity: antimicrobial cutting boards, toothbrushes, bedding. Public spaces are saturated with the scent of disinfectant. In some places, the ritual of hand-washing has been elevated to a symbol of civic virtue, a marker of personal responsibility and care for the community. This crusade against bacteria has its own ironies, its own unintended consequences, for in our zeal to cleanse we have sometimes overlooked the complexity of the world we inhabit. The good and the bad, so easily separated in cartoons and commercials, are in reality threads woven tightly together. The same act of sterilization that protects against disease may also disrupt the delicate balance of microbes that sustain our health. The war on germs, like all wars, breeds its own casualties. And so we find ourselves in a peculiar position, more aware than ever of the invisible kingdom that surrounds us, yet more anxious, more suspicious, more eager to banish it from our midst. We scrub and rinse and spray, chasing an ideal of purity that can never be fully attained. The bacteria, for their part, persist, adaptable, ancient, indifferent to our campaigns. They have lived on this planet for billions of years, through ice ages and extinctions, through the rise and fall of continents, long before the first human ever dreamed of a clean kitchen or a sterile operating room. The stories we tell about bacteria, then, reveal as much about us as they do about the microbes themselves. In their faceless multitudes we see both threat and opportunity, contamination and connection. We project our anxieties onto them, but also our hopes, for healing, for understanding, for mastery over the forces that shape our lives. The invisible kingdom becomes a mirror, reflecting the fears and aspirations of its human observers. And yet, if you lean in close, if you listen not just to the warnings and the alarms, but to the quieter voices, another story begins to emerge. It is a tale not of war, but of coexistence, not of eradication, but of balance. For though bacteria have been cast as villains in the dramas of our age, they are in truth far more complex and fascinating than these simple roles allow. Their world is not just a battlefield, but a tapestry of relationships, an ecosystem more intricate than any forest or coral reef. Consider, for a moment, the paradox. We fight so fiercely against bacteria, yet we are utterly dependent on them. Without them, there would be no soil, no digestion, no oxygen. The very air you breathe has been shaped, molecule by molecule, by the labor of ancient bacteria. The food you eat, the health of your body, the cycles of birth and decay, all are touched by their unseen hands. Even as we wage our wars of cleanliness, we are in every moment hosts to trillions of bacterial guests. They line our skin, fill our mouths, colonize our guts. We are, in a sense, walking ecosystems, each as unique and dynamic as any rainforest. But this is not the story we tell in our movies or our advertisements. There, bacteria remain the enemy, the poison, the threat. The invisible kingdom is a place to be conquered, not explored. Its denizens, objects of fear rather than curiosity. We have, perhaps, inherited this view from the earliest days of germ theory, when the discovery of unseen life was met with a mixture of awe and terror. It is easier, after all, to fear what we cannot see than to embrace it. And yet, there are cracks in the narrative, hints of a deeper truth. In recent years, a new appreciation has begun to grow, a recognition that the invisible kingdom is not just a source of peril, but of possibility. The language of microbiome has entered the public consciousness, and scientists now speak of good bacteria with the same reverence once reserved for vitamins or antioxidants. Probiotic yogurts line the shelves, promising better health through microbial partnership. The story is changing, slowly, unevenly, but unmistakably. Still, the shadow of fear remains. The invisible kingdom is vast, its workings mysterious. For every tale of beneficial bacteria, there is another of outbreak and contagion. We are caught between two impulses, to cleanse and to embrace, to exclude and to welcome. The bacteria, meanwhile, go about their ancient business, shaping the world in ways we are only beginning to understand. In the hush of night, as you lie in the quiet of your room, consider this. You are surrounded, inhabited, sustained by an invisible kingdom, older than any story, more wondrous than any fiction. And though we have cast its inhabitants as villains, they are in truth our oldest companions, mysterious, adaptable, and essential. The stories we tell about them are just the beginning. Their true nature waits, patient and persistent, for us to look again, to see beyond the shadows, to listen to the whispering world that lies just beneath the surface. And so let your thoughts linger at the border of this unseen realm, poised between fear and fascination, as the story of the invisible kingdom unfolds quietly in the dark. Beneath the surfaces we see, beyond the clarity of glass or the smoothness of our skin, lies a realm so intricate, so teeming, that it dwarfs the grandest metropolises of human imagination. This is the world of bacteria, where complexity is woven into the very fabric of the minuscule. Here, in the domain of the infinitesimal, the curtain lifts on a stage that hosts the oldest, most adaptable, and perhaps most misunderstood actors in the theatre of life. Let us linger at this scale, where a single droplet of pond water becomes a universe, animated by countless forms, most of which will never draw the eye of a passing heron or the curiosity of a child. At this level, the myth of the malevolent bacterium, that simple persistent tale that all bacteria are harbingers of disease, quickly dissolves. It gives way to an altogether more nuanced story, one that reveals bacteria as essential architects and caretakers of life. If you could shrink yourself to the dimensions of a bacterium, you would find yourself adrift in an ocean of chemical signals, awash with opportunity and peril. You might notice the way these creatures cluster in slimy colonies, communicating through whispered molecules, or the way they dart about, powered by whirring flagella, tiny corkscrews of protein that rotate with an efficiency unmatched by any human invention. You might brush past a cell wall studded with proteins and sense the molecular machinery within, tirelessly copying DNA, repairing damage, crafting enzymes, or ferrying nutrients across the membrane. And if your senses could stretch to the limits of the possible, you would hear the faint chorus of genes being switched on and off, a symphony of survival and adaptation. But perhaps the most profound revelation would come from realizing how dependent all larger life forms, including ourselves, are upon these nearly invisible beings. The story of bacteria is not merely one of survival or proliferation. It is a story of symbiosis, of mutual dependence, and of an ancient, intricate network of relationships that underpins the living world. Consider, for instance, the constellation of bacteria that makes its home within your own body. Even as you lie comfortably in your bed, your skin, your mouth, your gut, and every moist crevice harbour thriving communities of bacteria. These are not passive stowaways, they are active collaborators, shaping our biology in ways both subtle and profound. Collectively, this inner ecosystem is known as the human microbiome, and it contains, by some estimates, as many bacterial cells as there are human cells in your body. Inside your intestines, for example, reside trillions of bacteria, representing hundreds of species. They form a living, shifting tapestry along the folds and villi of your digestive tract. Their presence there is no accident. Over millions of years, a fragile truce has been negotiated. The bacteria help us break down complex carbohydrates and fibers that our own enzymes cannot digest, extracting nutrients and producing vitamins like K and B12, while we in turn provide them with warmth, moisture, and a steady supply of food. This is not a static relationship, but a dynamic, ever-evolving one. The composition of our gut microbiome shifts with our diet, our age, our health, and even the seasons. This partnership is so fundamental that it shapes not just our nutrition, but also our immune system, our metabolism, and even, indirectly, our moods and behavior. Signals from gut bacteria can influence the development of immune cells, teaching the body to distinguish friend from foe. Some bacteria produce short-chain fatty acids that nourish the cells lining the colon and help regulate inflammation. Others manufacture neurotransmitters or modulate the production of hormones, sending messages that ripple from the gut to the brain, a pathway that some scientists have dubbed the gut-brain axis. It is a reminder that the boundary between self and other, between human and microbe, is far blurrier than we once believed. Yet, for all this intimacy, the world of bacteria remains largely hidden from us, both physically and conceptually. The vast majority of bacterial species are strangers, never cultured in a laboratory, never named or mapped. Their genetic blueprints, recovered by scraping soil, swabbing the hulls of ships, or filtering seawater, hint at abilities and metabolisms that challenge our imagination. Some thrive in boiling springs, others in frozen tundra or the crushing depths of the ocean. There are bacteria that breathe metals instead of oxygen, that feed on oil slicks, or that precipitate minerals into delicate crystalline patterns. There are even bacteria that harvest energy from sunlight, not with green chlorophyll, but with purple or orange pigments, painting the world at a scale too fine for the unaided eye. This hidden diversity is matched by an adaptability so profound that it borders on the miraculous. Bacteria have endured every catastrophe the planet has thrown at them: fire and ice, poison and drought, the rise and fall of continents, the waxing and waning of oxygen itself. Their secret is their simplicity. Each bacterium is a single cell, small enough to replicate quickly, flexible enough to swap genes with neighbors, and tough enough to hunker down in the most inhospitable of places. When conditions sour, some bacteria form spores, tiny armored seeds that can survive boiling, freezing, even the vacuum of space. When food is scarce, they slow their metabolism to a crawl, waiting years, decades, or centuries for the world to change. But adaptability is not merely a matter of endurance, it is also a matter of ingenuity. Bacteria communicate with chemical signals, a language known as quorum sensing, which allows them to coordinate their actions as a group. When enough bacteria gather, their signals reach a threshold, prompting them to change behavior, perhaps to form a biofilm, a slimy community that clings to rocks, teeth, or medical devices. Within these biofilms, bacteria are shielded from antibiotics and immune attacks, sharing resources and information, huddled together in collective defense. This cooperative spirit extends beyond their own kind. In the soil beneath our feet, bacteria form alliances with plants, exchanging nutrients in a silent commerce that sustains forests and fields. Nitrogen-fixing bacteria, for example, burrow into the roots of legumes, converting atmospheric nitrogen, a form inaccessible to most life, into ammonia, a nutrient plants can use. In return, the plant supplies the bacteria with sugars and shelter, a partnership so ancient that the blueprints for it are encoded in both genomes. Without such collaborations, the cycle of life would falter, crops would wither, and the green revolution that sustains humanity would never have taken root. In the oceans, bacteria are the unseen engines of the biosphere. They drift in the plankton, breaking down dead matter, recycling nutrients, and forming the foundation of the food web. Some, known as cyanobacteria, are the architects of the air itself. Billions of years ago, these blue-green bacteria unlocked the secret of photosynthesis, splitting water to harvest energy from sunlight and in the process releasing oxygen as a waste product. They transformed the planet's atmosphere, paving the way for the evolution of animals, including humans. Even today, cyanobacteria and their descendants generate more than half the oxygen we breathe, their invisible labor sustaining the breath of all creatures. Yet, despite this bounty, the relationship between humans and bacteria is not always harmonious. The balance can be tipped, alliances can fray. When our immune defenses are weakened, or when bacteria find their way into places they do not belong, blood, spinal fluid, the sterile recesses of a surgical wound, they can turn opportunistic, causing infection and disease. Some species have evolved elaborate mechanisms to evade our immune system, cloaking themselves in slippery capsules or secreting toxins that sabotage our cells. They multiply with astonishing speed, outpacing our defenses and in some cases resisting our most potent antibiotics. But even here, in the realm of disease, the story is more complex than it first appears. Many pathogens are not inherently malevolent. They are simply bacteria in the wrong place at the wrong time, or creatures whose survival strategies conflict with ours. The vast majority of bacteria, even those with the capacity to cause harm, live their lives in peace, playing essential roles in decomposing waste, cycling nutrients, or maintaining the health of ecosystems. Indeed, the very term bacteria is an umbrella sheltering an astonishing diversity of forms and lifestyles. There are bacteria shaped like rods, spheres, spirals, and filaments, some solitary, others forming chains or branching networks. Some possess multiple membranes, others just one. Their genomes are mosaics, constantly shuffled by the exchange of plasmids, small rings of DNA that can carry genes for antibiotic resistance, toxin production, or novel metabolic tricks. In the collective library of bacterial DNA lies a record of countless evolutionary experiments, some successful, others fleeting, and still our understanding is rudimentary. Every advance in technology, from the invention of the electron microscope to the sequencing of genetic material, has revealed new mysteries. We have learned that bacteria can transfer genes not only to their offspring, but sideways to unrelated species, a process called horizontal gene transfer, which allows them to adapt with breathtaking speed. We have glimpsed viral invaders, known as bacteriophages, that prey upon bacteria, shaping their evolution in a microscopic arms race. We have discovered bacteria living in the clouds, seeding raindrops, and in the crust of the earth, kilometers below the surface, sustained by chemical energy alone. This is the complexity of the minuscule, a world where scale belies importance, where cooperation and competition, creation and destruction unfold in endless cycles. The more we learn, the more we realize how little we truly know. The bacteria that share our bodies, our homes, and our planet are not mere background players. They are the silent majority, the ancient custodians of life, and the architects of worlds both visible and invisible. As you rest tonight, consider that every breath, every mouthful, every heartbeat is shaped by the unseen. The boundaries between you and the minuscule are permeable, shifting, and alive. Bacteria are not merely agents of disease, but partners in existence, bearers of diversity and guardians of cycles that began long before we arrived and will continue long after we are gone. The night deepens, and in the darkness, the bacterial world thrums on, ceaseless and unseen. Its secrets are many, its wonders without end. Beyond the complexity of the cell, deeper questions beckon. How did this world arise? What forces shaped the first bacterial life? And how do the patterns of the minuscule echo in the grand sweep of evolution? These are the mysteries that await us, carried forward on the tide of a story that began with the simplest spark, in a world unimaginably small but infinitely rich. In the hush of a pre-dawn laboratory, where the soft clink of glass slides and the faint hum of anticipation hang in the air, a lone observer might pause before a curious contraption. A handful of glass lenses, carefully arranged in a brass tube, trembling on the edge of revelation. It was the 17th century, and the world was about to open its eyes to a secret kingdom that had always been there, quietly shaping the fate of everything it touched. The microscope, simple in its first incarnations, yet profound in its implications, was the key to a door long closed. Antony van Leeuwenhoek, a Dutch tradesman with an eye for the infinitesimal, bent over his handcrafted lenses and peered into the invisible. He was not a scientist in the formal sense, no degree, no laboratory, only an insatiable curiosity and a patient hand. Yet, with every careful twist of the focus, Leeuwenhoek found himself staring at a riotous world. Droplets of pond water teeming with animalcules, tiny creatures darting and spinning, so alive, so intricate, so very beyond the limits of ordinary sight. He drew them, He counted them. He marveled at their forms. Some were rod-shaped, others round or spiral. He saw what no one had seen before, and in doing so he shattered the illusion that the world was made only of what the eye could see. Leeuwenhoek's letters to the Royal Society in London caused a sensation. Learned men argued, some in disbelief, others in awe. What could these invisible creatures mean? Were they the seeds of disease, as some would later suggest, or simply another form of life woven into the fabric of existence? The microscope, humble yet transformative, did not answer these questions outright. Instead, it beckoned generations of explorers to peer deeper and to ask better questions. For centuries, the microscope was both a window and a barrier. Its glass brought distant worlds close, but it could not explain what it revealed. It took the insights of men like Robert Koch and Louis Pasteur to begin connecting the teeming world seen under the lens with the mysterious processes that governed health and sickness. In the dim lamplight of 19th century laboratories, glass slides multiplied, and stains and dyes were invented to tease out the forms of bacteria from the murk of organic matter. The Gram stain, devised by Hans Christian Gram, turned invisible cells into purple and pink banners, classifying bacteria into two great kingdoms, Gram-positive and Gram-negative. With each improvement, the code of microbial life became a little less cryptic, yet while the microscope let us see, it could not tell us how these tiny creatures lived, or what powers they held in their minuscule hands. This was a mystery best unravelled by experiment, by the careful dance of hypothesis and test, of failure and epiphany. Consider for a moment a forgotten dish of culture medium, a simple petri dish left uncovered on a cluttered workbench in a London hospital laboratory. It is the year 1928, and Alexander Fleming, weary from work, is tidying up his experiments on staphylococci. He notices, almost by accident, that where a stray mould has landed, the colonies of bacteria have vanished. The mould, it turns out, is Penicillium notatum, and it exudes a substance that kills bacteria but leaves human cells unharmed. Fleming has stumbled upon penicillin, not the first antibiotic, as nature has been at this game for millennia, but the first to be recognized, purified, and harnessed by humankind. The discovery of penicillin was not the end of a story, but the beginning of a new chapter in deciphering the microbial code. It was a lesson in serendipity and in the ceaseless dialogue between humans and microbes. For even as penicillin and its kin vanquished infections that had long haunted humanity, bacteria answered back. Mutations, plasmids, and gene swaps gave rise to resistance, a reminder that the microbial world is not a static foe, but a living, evolving system, one that listens, learns, and adapts. But how do bacteria adapt? What is the machinery that lets them change, that lets them survive the onslaught of our cleverest drugs? To unravel this, scientists needed to look not just at shape or color, but at the very instructions that guide a bacterium's life, the genetic code. It was at this threshold, in the middle of the 20th century, that a new set of tools emerged, the tools of molecular biology. The world's attention turned to the winding helix of DNA, that double-stranded ladder carrying the blueprints of every living cell. The bacterium Escherichia coli, or E. coli, became the model system, a laboratory favorite, not because it was the most exotic, but because it was tractable, swift to grow, easy to manipulate, and representative of the wider bacterial domain. The simplicity of E. coli belied its power as a window into the secrets of life. In the hands of researchers like Matthew Meselson and Franklin Stahl, E. coli revealed the very mechanism of DNA replication. In their now classic experiment, they fed bacteria a diet of heavy nitrogen, which became incorporated into the DNA. Then, by shifting the bacteria to a diet of light nitrogen and observing the density of DNA after successive replications, they demonstrated that DNA replication is semi-conservative. Each new strand contains one old and one new half. It was a revelation of elegance, showing that the code of heredity is copied with both fidelity and flexibility, a mechanism as precise as any clock, yet open to the subtle shifting of mutation. Other experiments equally clever followed. The famous lac-operon experiments of François Jacob and Jacques Monod illuminated how bacteria regulate their genes. They showed that E. coli, when faced with a choice between sugars, could turn certain genes on or off in response to the presence of lactose. This was no random process. It was a tightly controlled logic circuit, a molecular decision-making system. The genes for digesting lactose were kept silent, locked away by a protein repressor until lactose itself appeared and bound to the repressor, freeing the genes to be expressed. Here was a code within a code, a living program, written not in silicon but in molecules. As the 20th century unfurled, the tools for deciphering this code grew ever more refined. The electron microscope, with its torrents of electrons and powerful magnets, allowed scientists to see bacteria in stunning detail. Membranes, flagella, even the delicate machinery of ribosomes, where proteins are spun from the threads of RNA. No longer were bacteria mere dots and rods. They were intricate machines, each part whirring in concert, each process governed by the orchestration of genetic information. The dawn of genetic engineering brought a new alchemy. Restriction enzymes, discovered in bacteria themselves as a defense against viruses, became scalpels for cutting DNA with precision. Scientists could snip, splice, and recombine genetic material, creating recombinant DNA and ushering in the age of biotechnology. E. coli became a workhorse for producing insulin, growth hormones, and vaccines, a humble bacterium pressed into the service of human health, its genetic code rewritten for novel purposes. Yet, as powerful as these tools were, they could not reveal the whole story. The microbial world is not a collection of isolated actors, but a churning, interconnected web. To understand it, scientists needed to look beyond single genes or solitary cells. They needed to see the orchestra, not just the individual players. The emergence of high-throughput DNA sequencing provided that panoramic view. The sequencing of entire bacterial genomes, first Haemophilus influenzae, then E. coli and countless others, exposed the full breadth of genetic diversity. Genes for antibiotic resistance, for virulence, for metabolism, all could be mapped, compared, and traced across time and geography. Scientists began to speak of the pangenome, the full set of genes within a species, not just in one strain, but in all its myriad variants. The microbial code, it turned out, was not static. It was a living text, written and rewritten by evolution, by the horizontal transfer of genes, by the ceaseless churn of mutation and selection. With each new technique, from polymerase chain reaction, PCR, to CRISPR gene editing, the veil over the microbial world slipped a little further. PCR, devised by Kary Mullis, allowed the amplification of tiny fragments of DNA, an echo of how bacteria themselves copy their genetic material, now harnessed by humans to detect pathogens, identify genetic fingerprints, and explore the depths of microbial diversity. CRISPR, borrowed from the immune systems of bacteria, became a tool for precise genetic editing, a molecular scalpel guided by RNA. But among all these dazzling innovations, perhaps the most profound shift was not in what we could do to bacteria, but in what we could learn from them. Bacteria are the consummate survivors, the master engineers of the genetic world. They swap genes with abandon, picking up new tricks from viruses, from other bacteria, from the environment itself. They store fragments of viral DNA in their genomes, a molecular memory of past invasions, ready to be called into service should the same foe return. The code they write is not only for themselves, but for their descendants, for their kin, for the entire microbial community. And so the history of microbiology becomes not just a chronicle of tools and techniques, but a story of dialogue, a conversation across the centuries, across the gulf between human and microbe. Each experiment, each discovery is a line in this ongoing script, a step closer to understanding how the smallest forms of life shape the largest patterns of existence. In the gentle glow of lamplight, as researchers bend over their benches, a kind of reverence settles in. They are not merely dissecting life, but communing with it, deciphering a code that was written long before the first microscope, a code that will continue to evolve long after the last experiment has ended. Through the lens, through the petri dish, through the whirring hum of sequences, the microbial world whispers its secrets. Some are stark and urgent, the rise of resistance, the threat of new pandemics. Others are subtle, almost poetic, the dance of genes, the quiet persistence of life in the harshest corners of the earth. Yet, for all we have learned, the code remains, in many ways, unfinished. There are genes in bacterial genomes whose functions we do not yet understand, stretches of DNA whose roles are known only to the bacteria themselves. New species are discovered not by sight, but by sequence, a fragment of DNA in a scoop of soil, a trace of life in the depths of the ocean, a pattern in the air we breathe. This is the frontier now, not just the world seen under the microscope, but the hidden realms revealed by the code. A spoonful of earth, a droplet of seawater, a breath of air, all teem with microbial life, woven together by the threads of DNA, each sequence a story, each gene a possibility. The tools of modern microbiology let us read these stories, trace their connections, glimpse the vastness of the microbial universe. The journey, it seems, is far from over. For as we peer deeper into the code, as we decipher the patterns and translate the language of bacteria, new questions arise. How do these microbes shape the world beyond themselves? How do their communities form, fragment, and reform? What secrets do they hold about the past? And what hints do they offer about the future? The answers, like the microbes themselves, wait quietly, hidden in plain sight, their code ready to be read by those willing to look, to listen, to learn. In the next quiet moment, as the laboratory falls still and the world slips toward sleep, the microbial code continues to write itself, an unfinished manuscript, waiting for the patient hands and curious minds who will turn its next page. In the hush of a darkened room, as sleep begins to soften the edges of waking thought, let us turn our inner gaze to the world that so often escapes our notice, a world not above or beyond us, but within and alongside us, so intimately entwined with our existence that the boundaries blur, and we are left to wonder, where does the human end and the bacterial begin? Bacteria. The word itself conjures a multitude of images, sometimes fear and revulsion, sometimes fascination, and for those who look deeper, a kind of reverent awe. They are the smallest architects of the living world, the invisible laborers at the foundation of all ecosystems, the quiet sustainers of cycles that have spun since the dawn of life. To reflect on our relationship with these minute beings is to enter a hall of mirrors where every reflection reveals something new, something unsettling, something profound about what it means to be alive. Close your eyes, if you wish, and let your mind expand beyond the surface of your skin. Imagine for a moment the entire population of bacteria that calls your body home. There are more bacterial cells on and within you than there are human cells, by some estimates 3 times as many. Each millimeter of your skin, each fold of your intestine, each moist crevice and arid plain of your body is a habitat, a metropolis, a thriving city-state for multitudes of species. They are not invaders, nor are they mere passengers. These bacteria are partners, symbionts, sometimes even guardians. The story of you is in a very real sense a story of them. Let us listen to the bacterial symphony, not with our ears, but with the full attention of our imagination. Within your gut, a choir of trillions tunes itself to the rhythms of your diet, your mood, your age. They digest the fibers you cannot, fermenting them into short-chain fatty acids that nourish your colon and, indirectly, your brain. Certain bacteria produce neurotransmitters, yes, the very molecules that modulate thought and feeling, such as serotonin and dopamine. Others refine vitamins and harvest energy from compounds you cannot access alone. Some are ancient friends, passed down from mother to child, generation upon generation, in a lineage as old as humanity itself. But the relationship is more profound still. Imagine each breath you take carrying with it invisible passengers, airborne bacteria, spores, and fragments of microscopic life, settling onto your lungs and mingling with the communities already there. Every meal, every touch, every embrace is an exchange, a gentle mingling of microbiomes. Even now, as you rest, your body is a bustling, humming marketplace of chemical exchange, negotiation, and cooperation. To the philosophers of old, individuality was a clear and simple concept. I am me, and you are you. But the bacterial perspective dissolves this tidy boundary. The human is revealed as a consortium, a walking, thinking superorganism, composed not only of the 23,000-odd human genes nestled in our chromosomes, but also of millions of bacterial genes, each with its own story, its own function, its own ancient lineage. Our immune system, once thought to be a vigilant defender against all foreignness, is now understood as a diplomat, a negotiator striving for harmony among the multitude of microbial citizens within. In this light, the self is not a fortress, but a city, open, dynamic, ever-changing, always in dialogue with the unseen. What does it mean, then, to be human? Are we the sum of our parts, or something more? The answer, it seems, is both humbling and ennobling. We are not isolated islands, nor are we mere hosts for passengers. We are, instead, symbiotic collectives, each of us the result of a billion-year experiment in cooperation, competition, and coexistence. Consider the story of the mitochondrion, the power plant of the cell. It too was once a bacterium, free-living, independent, perhaps even predatory. Over 1 billion years ago, it entered into a partnership with an ancestral cell, trading autonomy for sanctuary, offering energy in exchange for protection. That ancient merger was so successful, so vital, that it was passed on to every eukaryotic cell, becoming the engine that powers all higher life. Even today, the mitochondria in your muscles, brain, and heart retain traces of their former independence, their own DNA, their own peculiarities. In every breath you take, every thought you think, the legacy of bacterial partnership endures. But our relationship with bacteria is not only a story of cooperation. There are tensions, competitions, and yes, conflicts. Some bacteria, under the wrong circumstances, can turn against us, causing illness, inflammation, or worse. Yet even here, the story is more nuanced than it seems. Many pathogens are not alien invaders, but familiar residents thrown out of balance by antibiotics, stress, or dietary change. The lesson, perhaps, is not simply to eradicate, but to restore harmony, to seek equilibrium rather than war. Our culture, for generations, has been shaped by the war metaphor. We fight infection, battle disease, sterilize, and disinfect. Yet as we peer deeper into the bacterial symphony, a different image emerges, not of endless conflict, but of a delicately tuned orchestra where each player has a part to perform. The immune system is not merely a wall, but a conductor, guiding the tempo, keeping the peace, allowing the soloists their moments of glory while quieting the discordant notes. This concept of the human as ecosystem, as symphonic ensemble, has begun to reshape medicine, nutrition, even philosophy. Probiotics and prebiotics, once the province of fringe science, are now recognized as potent tools for restoring microbial balance. Fecal transplants, a procedure that once provoked disbelief, have become a lifeline for those with intractable infections. Researchers speak in terms of microbiome health, recognizing that well-being is a property of the whole, a harmony, not a single note. There is, too, a strange kind of democracy in the microbial world. No single species rules for long. Dominance is fleeting, balance is everything. When antibiotics kill off a swath of bacteria, others rush in to fill the void, sometimes for better, sometimes for worse. When diet or environment changes, so too does the makeup of the microbial community. The body is not a fixed entity, but a landscape in flux, ever responding, ever adapting, shaped by the tides of daily life. Let us contemplate for a moment the notion of agency. When you crave a certain food, is it your conscious self desiring it, or are the bacteria in your gut subtly nudging your preferences, shifting your mood, even influencing your choices? The idea feels almost heretical, yet recent research suggests that microbial metabolites can cross the blood-brain barrier, altering neurotransmitter levels, affecting appetite, even modulating anxiety and depression. To see ourselves as wholly autonomous beings is perhaps an illusion, a comforting story told to us by the conscious mind, while beneath the surface 1,000 voices whisper in chemical tongues. This interconnectedness extends beyond the individual. Our bacterial symbionts are not passed down solely through inheritance, but through community, through shared meals, physical contact, the air we breathe together. Families, friends, even strangers on a crowded train share more than air. They share a microbial milieu, a cloud of invisible life that drifts from person to person, knitting us together in ways we rarely consider. And so the boundaries of self begin to dissolve. The I becomes we, a chorus of human and microbial voices rising and falling in endless counterpoint. The philosopher's ancient question, what is the self, takes on new resonance. We are not monoliths, but collectives. Our bodies are symphonies. Our minds, perhaps, are shaped by the music of unseen players. There is humility in this realization, but also a quiet joy. To know that we are not alone, that our existence is supported by a host of ancient allies, is to see the world with new eyes. No longer are bacteria mere background noise, but essential partners, their presence a reminder of the deep interdependence that underlies all life. The earth itself is a vast living tapestry of microbial interactions, from the soil beneath our feet to the clouds above, from the deepest ocean trench to the lining of your smallest capillary. Pause for a moment and consider the scale of this invisible world. A single gram of soil contains billions of bacteria, outnumbering the stars in the Milky Way. Each leaf, each drop of seawater, each grain of sand is a universe in miniature, teeming with life. Bacteria are the great recyclers, breaking down dead matter, fixing nitrogen, breathing metals, and ensuring that the cycles of carbon, sulfur, and phosphorus continue unbroken. Without them, the world would grind to a halt, life as we know it would collapse. Yet for all their ubiquity, bacteria remain mysterious. The vast majority cannot be cultured in the laboratory. Their genomes, only recently accessible through the miracles of sequencing, reveal a staggering diversity of function and form. Some are so alien in their metabolism, so strange in their molecular machinery, that they force us to reconsider the very definition of life. What else, we must wonder, waits to be discovered in the unseen depths of the microbial world? Perhaps, as you drift toward sleep, you might sense the gentle reassurance of this connectedness, the knowledge that you are never truly alone, that your every breath and heartbeat is accompanied by a silent chorus. The bacteria within you are not only keepers of health, but also custodians of memory. They remember the foods you have eaten, the places you have lived, the illnesses you have endured. They adapt, learn, and persist, passing on their wisdom to the next generation, shaping not just your body, but the bodies of those who come after you. This is the quiet magic of symbiosis, the realization that life is not a solitary endeavor, but a collective enterprise. Each human, each animal, each plant is an ecosystem, a cathedral built from countless lives interwoven. To care for oneself is, in a sense, to care for this community, to nurture, to balance, to honor the invisible threads that bind us to the microbial world. As the night deepens, let your thoughts wander to the future. What new partnerships might we forge with our bacterial companions? Can we learn to cultivate harmony, to restore what has been broken, to heal not just ourselves, but the living earth? The questions linger, unanswered, like notes suspended in the air after the last chord of a symphony. And in that silence, in the darkness before dream, the bacterial symphony plays on, a rhythm older than memory, a melody that pulses through every living thing, inviting us to listen, to wonder, and to join in the music of life unending. As our journey through the secret life of bacteria drifts to a close, we float gently like a comet tail fading into the hush of space, marveling at the invisible worlds that quietly run our earth. May this quiet wonder settle softly in your mind, inviting peaceful dreams and gentle curiosity. Until we meet again beneath the stars, return for more Dreamtime tales that illuminate the mysteries whispered by the cosmos.